Okay, so I've added a link, and you can even see it on your front page. When you click on it, make sure you answer all of those. And then you see right here it says submit assignment. So you could like type this up in Word or whatever, and then you click submit assignment, and you could just paste and copy it in here, or you can write it in here. I always caution students not to do that when it's like a long thing that you're writing up, because if you lose internet connection depending on where you're doing this, like anything you type in these boxes doesn't save, right? So learn from, you know, other people's problems of typing in a thing that doesn't automatically save. Don't do it if you're typing a lot, right? I've done this before, needless to say, right? And then you lose what you typed. So I would type it up somewhere else and then just paste and copy it in here and then hit submit to submit it. So, so you need to give me your proposal by next Tuesday. You can work in groups, but you guys got to get together, write up the proposal together, and everybody submit the same proposal, right? And of course, obviously, write in there, you know, everybody's names in that group. The max I would allow is 3 people per group. Okay. And at some point too, I'm going to want it divvied out who did what. I'm not going to make you guys necessarily do that ahead of time, but I don't think I really have to worry about that with you guys, but for grading purposes. Okay. So that's my student view. So if you saw my announcement, right, I don't know about you guys, I was reading over chapter 8, and yeah, it was like just like basically a whole chapter worth of terms, right? It was like insanity. I was like, okay, for right now I'm definitely skipping this. We might be omitting it altogether, but I think it's got some stuff in it that we're required to cover. So I'm going to find some way to tease out what it is we have to do, and that's all we will do, if anything. So I decided to go ahead and jump ahead, as I said in my announcement, to Chapter 9. So that's what we have for today, and I had posted that for you guys. So Chapter 9 is pretty awesome, because we start molecular genetics, right? So at the molecular level. Yep, I'm kind of the only one that gets excited about that right now. Okay, what's the good news? The good news is you guys probably know a lot of this information, which reminds me, I wanted to do something today. I need a volunteer, because it won't let me do it, that knows their, at least their password to Lola. Anybody want to volunteer? I just need to pirate your account for a second. Okay, so I need a student account. I want to show you guys something.
My email is different now.
Oh, I know. I had to change mine too. It's driving me crazy. Got it. Make sure the caps lock isn't on. Okay.
Oh, that's fine.
Yes, thank you. I won't save it. Okay, so when you guys go into Connect, which actually, okay, I'm not going to read all of that right now. Oh, you guys do see the assignment list. Oh, did not know that. Look, I already put it, but I didn't actually assign it to you guys yet. I didn't think I did. Maybe I did. I just didn't post it in Canvas. Okay, so when you click on the book, but if you click on your library and you click on student resources, the reason why I can't show you guys because it says instructor resources because I've got more stuff than you guys. and then you choose a chapter, you'll see that they have little quizzes and stuff, right? So here's a multiple choice quiz for this chapter. So let's see, how many questions is it? I don't really care about the true and false. 15. Let's go through the first 10 and see what you guys know or don't know, or a few of them. Let me see. Okay. Is it this tiny, huh? Let's make it bigger. Okay. All right. Let's see how much prior knowledge you guys have. So which of the following is a key component of a DNA nucleotide? Anyone disagree? Right? That was an easy one. What's the difference between purines and pyrimidines? Ugh. Do you even know what a purine or a pyrimidine is? Is it cut? No, it's not cutting it off. Okay, so let's read through them. So purines are found in RNA while pyrimidines are found in DNA. Anyone think that's true? Anyone know what purines and pyrimidines are referring to? What part of the nucleotide? The nitrogenous bases are put into two classifications, purines and pyrimidines. So the next one is very similar, right? They just switched it. Is that one true? No. No, right? Both are found in both RNA and DNA, as we'll see as we go through the lecture. So purines contain a double ring background, while pyrimidines contain a single ring background. You think that one's it? Okay. And then the other one is just the opposite of that, right? Is pyrimidines are double ring and purines are single ring. So if you had me for microbiology, I actually gave you guys a tip for remembering this. But you don't need to know it unless you're in this class. Purines have two rings. And how I remember this is a little saying that I learned, is that angels are pure and angels have two wings, which rhymes with rings, which you start rhyming and the words all start running together, right? So here, I'm just going to add it here, but I'll put it where it belongs later. So pure, right, makes you think of purines. Which makes you think of the rings. Right? It's a play on words, right? What did I spell wrong? Oh, two. The number two, not two. Yes, yes. So did you see? Like a play on words. Purines for pure, right? And wings for rings. So that was a saying that I learned that helps me remember each one. So when we actually look at the structure, right, they have two rings. So cool. Angela's got some prior knowledge that is working for her.
I remembered it because pyrimidine is just a long word that's even smaller than
Okay. Whatever works, right? What type of bonds connect the nucleotides? Yeah, it's phosphodiester. Because it's the phosphates and the sugars that interact with each other. The nitrogenous bases across the strands are hydrogen bonds. Which of the following statements about the structure of DNA is incorrect? Yeah, that one goes too specific. We're not going to go into that one just yet. What functional role is the three-dimensional structure of DNA likely to accomplish? So very long molecules of DNA can be packaged into small space. Information within DNA can be made available to drive cellular functions. Information within DNA is modified from generation to generation.
All of the above.
I think that one is all of the above. Yep, not going to worry about that one. Starts getting into too much specifics. But this is a good place to go. We're going to go ahead and submit our answers. All right, we didn't answer a lot of them. So let's see. The ones we answered is what we care about. So we got that one right, got that one right. Oh, it's A and B. Why is it A and B? I don't know about that one. Oh, no, it says incorrect. Oh, yeah, because it's saying A and B. Yeah, that's a tricky one. We'll have to think about that one. But we got that one right. That was the only other ones we answered right. So the only thing I'll caution you guys is if you were to use this, is notice down at the bottom, you can email, you can email yourself or you could add in my email. Do not do that. Okay? Good for you if you did it. I don't need to know you did it. But obviously if you have a question about it, right? Like we already have a question ourselves. I have a question about that one. I have to do some more reading about that. The thing is, it's not a specific enough question, I think, is the problem. So just to let you guys know, I wouldn't obviously ask you a question like that, right? I don't think it's specific enough. But connects the nucleotide. That would be one nucleotide to another. I would say phosphodiester. I don't know where they're getting hydrogen. So I'll have to look up that. I'll look into that. Or maybe they're wrong. It's possible. This type of bond into a linear chain. Yeah, but they're also not referring to... Okay, but they didn't say DNA or RNA. So, yeah, it could be both. We'll talk about it. Okay. Huh? There another one we got incorrect that we clicked off, that we checked? All right, so let's see. So, unanswered. So, correct, correct, incorrect. Oh, it's saying this one's incorrect? Oh, we said all of the above. Yeah, see, I don't know what they're... This is too vague a question, too, I think. So, I'm not going to worry about it. So... What is the functional role? What functional role is the three-dimensional structure of DNA likely to accomplish? So they're saying it functionally, but it is modified from generation to generation because we talked about imprinting, and that happens at the three-dimensional level when they modify the histones. Yeah. Yeah. I think so. I was a little too specific and yet not specific. Okay, so Shay, so I don't screw anything up. I'm logging out of your account. I just wanted to show you guys that resource. Right, so as you're studying, that's another, like if you need more questions, although you got plenty in the back of your book too. So let's actually, I was kind of hoping there were better questions there. for pre-questions. One of the other books that I use actually gives pre-questions. I was hoping for a pre-quiz, you guys, see what you knew beforehand. But a couple of you guys have some prior knowledge, which is good. We're going to build upon that. So now we're going to get into the molecular components. So yeah, for some of us that don't like structures of molecules and stuff like that, not so much fun. I think it's pretty cool though. But of course, although we're talking about molecular, it's still linked to what we've talked about with transmission and what we can touch upon as it relates to population genetics. These are all interlinked, even though we talk about them separately, right? They are linked to each other. So a lot of what we know as it relates to genetics today comes from knowing what we do know about the molecular structure of both DNA and RNA. Before we could even get there, right, we had to figure out these things because this isn't something that we can pull out a microscope and look at, right? At least it definitely wasn't some 100 years ago. So there were some really important paramount experiments that were done that helped us determine that DNA was the molecule of heredity, genetics, right? So for each one of these, you need to know the specifics of course, whose experiment it was and what they did. So I know we're going to get through the first two today. I know we're definitely, and even if we have time, I'm going to skip over the third one because what I want you guys to do is I actually want you guys to read this one before we discuss it. And the other thing is it talks about some stuff that, depending on your background in microbiology, you may not understand that well. So, see, Shay, I knew I wanted to stay in your account and I didn't, but that's okay. So, everybody has access to the ebook. So you know within the book, too, there are videos, right, that you can link right to instead of like your paper copy of the book, you know. It's paper, you can't click a button and watch a video, right? But the electronic version you can. But in this PowerPoint too, I embedded this one video that I think would be helpful for you guys to watch. It goes over how bacteria are infected by viruses, specifically the lytic cycle. So that's important in understanding the Chase experiment. So first off, what are the 4 criteria that genetic material must fulfill? So what do you guys think? What must it do to be considered genetic material? Must have pass along information. Right, to be copied, right? So we got to be able to copy it. It's got to be information based, right? We have to be able to give it to the next generation. So that's 3 of them. Right, we've got that. We've got information. We've got to be able to copy it, replicate it. We've got to pass it on, what we call transmission. One more. Evolve, change. It has to have variation, right? It has to have variation. So it must have all the information. We must be able to pass it from parent to offspring. Must be able to be copied. Those kind of go hand in hand, right? Replication and transmission. If you can't copy it, you can't pass it on. But also, it must be capable of change, of variation. And this accounts for why we see phenotypic differences. Why would it be bad if all of us were exactly the same? Yeah, we could potentially all get wiped out by something, right? Some disease, right? Some environmental situation could wipe us all out if we were all genetically exactly the same, right? Variation actually makes us more survivable. This is why it's worrisome too, right? As some of our animal populations get smaller and smaller, the genetic pool, right, that population gets smaller and smaller. And so there's less variability within that population, right, and less mutation happens. But that's sometimes, not enough. So you end up with what's referred to as inbred, right? And a lot of the defects you can't get rid of because they're bred in, right? Everybody's got that defect. So who are you going to mate with to get rid of that defect? There isn't anyone left.
I was actually asked about that inbred, and I said because there's so much can go wrong with it because the mistakes and stuff are amplified between the houses.
Yeah, so think about, some genetic diseases that we see in particular populations, like Jewish populations, right? They have a higher instance of Tay-Sachs because, again, they have that defect, and then they're breeding with people who have that defect. So you're just amplifying that defect. Where if you bred with someone else, less likely to pass on that defect. The same is true for sickle cell, right? Because in that case, it actually is advantageous to have one allele to be heterozygous for sickle cell when exposed to malaria. Right? But variation is very important, for sure. So the data that we have collected goes hand in hand with what Mendel had studied, right? We're not we're keeping true to what he found out, except we're probably examining it even further. We couldn't, though, figure out the structure of DNA, right, and some of the things that we know about it from just doing crosses. So it wasn't until scientists like Griffith and honestly, it's one of those things like where a mistake happened or he didn't set out to discover this, right? He was trying to do something else. He was trying to develop actually a vaccine against this bacteria. And in studying the bacteria, known there then as pneumococcal, we now know it as Streptococcus pneumoniae, causes a pretty serious form of pneumonia. And he found that there was some variation within that species, within that population of bacteria. He found that some of them were smooth in appearance, and the reason for this is they had produced this polysaccharide capsule, this outer layer. The bad news for us is this was the virulent strain. This is the one that caused disease. And the reason for it is that capsule helped it hide from our immune system. So when you inhaled that bacteria, your immune cells that would normally destroy it, recognize it and destroy it, couldn't recognize it because of that capsule layer. It was hard for them to detect it. So it would grow to large numbers, right, cause a really serious form of pneumonia. If not treated, you would die. And that's what would happen to the mice that he injected with this strain of Streptococcus pneumoniae. But then he found there was another strain that looked a little bit more rough in appearance, and it didn't have a capsule. When he injected that into the mice survived. Right? They were able to detect it without that capsule layer, and they were able to kill the bacteria. So we played around with different things. He found that if he heat killed, now he killed it, right? The bacteria that has a capsule, the smooth strain, obviously he killed the bacteria, right? So when he injected it into the mice, It didn't kill them. But again, remember, he was trying to figure out a vaccine, trying to figure out immunity. So, of course, the cells that he injected into the mouse were dead. It didn't cause an infection, and he could not get back any living bacteria from blood samples or any samples from that mouse to grow an artificial culture. The next thing he tried was combining the two things that didn't kill the mouse. The cells that were killed that had the capsule and the cells that didn't have the capsule that were alive. He combined those together and injected that into the mice. And lo and behold, the poor mice die. What the heck is happening here? So he takes samples, right? And what does he get back? He gets living, encapsulated, smooth type bacteria, Streptococcus pneumoniae. Well, we know, right? How is it that that smooth cells, those encapsulated cells, how do they know how to make a capsule? Where is that information stored? Very simply, their DNA, right? So yes, the smooth ones in their DNA, in their code, is the information on how to make a capsule, right? So what happened is that the rough ones picked up that information from the environment, from the smooth cells, incorporated in and learned how, from that information, how to make a capsule. Because of that, they were said to be transformed. So this is each one of the steps just outlined. I'm not going to read through that. So he coined this phrase, right? What we still refer to today, where if DNA from the environment enters into a cell and that cell changes, that's referred to as transformation. So of course he didn't know what was moving, right, from the smooth cells to the rough cells. He didn't know it was DNA. He had no idea what it was. But he knew something was moving, right? Some particle, right? Some entity that he couldn't extract or quite understand. So at that time, it was referred to as the transforming particle. So at that time, notice I said time. There's a reason why I left that slide. This was back in 1928. Okay, so this was a long time ago. You think it's even weird that they knew as much as they did?
Yeah.
I think it's pretty impressive as well.
Did they just like create the light bulb?
I don't know what the timeline is on that. Yeah. Oh yeah, I mean, some of this research was done before, electricity.
I mean, lots of them, their experiments were done in the 60s.
50s.
On the corn.
Watson and Crick is the structure of DNA.
Who's the lady that did the genetic mutation?
Barbara McClintock. The really long, like 1600s, with Van Leeuwenhoek and yeah.
The transformation maybe means that the DNA has come from the environment and it's something that occurs within.
Right. Yeah, it's an external source of DNA. So this meets all the criteria for genetic information, right? Variation exists, right? You had the smooth ones versus the rough ones. It replicated. It was transmitted to the daughter cells. So it wasn't until we actually had developed techniques to actually separate out the different molecules that make up cells did we key in that it was actually DNA. So it was Avery Lloyd and McCarthy. So they took his work, and they carried these experiments out in the 1940s. At this time, other scientists had been able to extract out and separate out different components of cells. Separate out DNA from RNA, from proteins, from carbohydrates, and lipids. So they prepared cell extracts. So they actually took the smooth strain of Streptococcus pneumoniae, right, and they extracted out the DNA. Now the problem with that is DNA all by itself in a cell, even in a bacterial cell. Is it just DNA that makes up our chromosomes? There's proteins, right, that the DNA wraps around. Right, that it was DNA based because they extracted out the DNA. But the problem is that depending on what purification system you're using, there could still be protein there and there could still be RNA there. So how are we going to get rid of protein? How are we going to get rid of RNA? Right, they use enzymes. So they used enzymes. So we know that there are enzymes, right? And again, at this point, they knew about them. And so they used enzymes to destroy each one of them, even DNA. So they treated with RNases. These are enzymes that destroy RNA specifically. Proteases that destroy proteins. And DNases that destroy DNA. So only the extract that contained pure DNA was able to convert from rough to smooth, right? So when they did their experiment, only the extract that contained intact DNA actually caused the transformation. If they treated it with RNase, right, it didn't stop it. If they treated it with protease, it didn't stop it. But if they destroyed the DNA, no transformation happened. Right? So here are your rough cells, the control. The extract of the smooth cells of DNA. They got transformation. If they treated the extract with RNase, right, transformation still happened because the RNA was still intact. The DNA was still intact. Right? If they disrupted the protein with proteases, transformation still happened. But again, the question is, well, did you actually destroy all the RNA? Did you actually destroy all the protein? Right? Were there some contamination there? But really what helps seal it is when they actually digested the DNA with DNase, transformation did not happen. Right? So if you disrupted that DNA, you chewed it up with an enzyme, broke it into a whole bunch of pieces, transformation did not happen. So this is pretty strong evidence at this point that, okay, it's the DNA, right? So we know these molecules exist, right? These different types of molecules, and it is the DNA that actually is the genetic material. So remember I said we'd skip over Hershey and Chase. I want you guys to read up on this. Even watch this life cycle. So we're going to talk about those results next time. We're going to come back to that. So we're going to skip ahead to nucleic acid. So what is nucleic acid? When I say nucleic acid, what do you think?
I said products in a nucleus.
Yeah, but what's nucleic acid? Yeah, so you're going more specific, very general. What are molecules that we refer to as nucleic acid? DNA and RNA. Linnell, speak up. DNA and RNA. Right? So DNA and RNA. So most of the time when we say nucleic acid, though, most people think of just DNA, right? Because again, it's pretty darn important, right? It is our genetic code. It's our information. But not to be forgotten is RNA, right? Although poor little RNA is at the end of this chapter, right? DNA gets the big stage. So it's very interesting. I want you guys to actually read in your book. We'll see how many people read next time. How the name nucleic acid came to be. So in the first section, right, of section 2 in chapter 9, it tells you actually how they came up with the name nucleic acid. So the 4 levels of complexity is the nucleotides themselves, right? Then we're going to go into the specifics of what makes up a nucleotide. And then the nucleotides hook together to form what we call strands. And then the DNA we know is double-stranded in the form of a helix. That's the 3rd level. The 4th level is the 3D structure that it forms when it complexes with the proteins, and that's what we call chromosomes. Right? So each individual nucleotide, and then they form strands, and then the 2 strands can double helix, and then you can have the 3-dimensional structure, right, that gives rise to the chromosomes. So now when we start talking about a nucleotide, right, so you think what the main 3 parts? What do all nucleotides have, whether it be DNA or RNA? What do you think of when I say nucleotide? A sugar. What sugars? There's 2 different types of sugar. Anyone know the name of the sugars? Ribose and deoxyribose, right? Because that's where DNA gets its name, right? It's deoxyribo and RNA is ribonucleic acid. So we have a sugar, and we already said that that sugar binds with what to the next sugar, and so on. The phosphate groups. And then what actually gives the information is we call it what? What is an A, T, G, C, U? Adenine, thymine, uracil, guanine. They're the nitrogenous bases. Aha, it's important. The name for those is a nitrogenous base. And you do need to know that A stands for adenine, T for thymine, C for cytosine, U for uracil. Yep, you're in genetics now. You actually have to know what the letters stand for. Okay. And so what are the differences versus DNA and RNA? So we already talked about one of them, right? Is the sugar. Which one does RNA have? Ribose. Which one does DNA have? The deoxyribose. Okay. Nitrogenous bases. Are they all in DNA? No. Is A? Yes. Adenine, yes. T?
No.
DNA? Yes. T's are in DNA. Thymines are in DNA.
I thought you were talking about RNA.
Huh? Right. Uracil is not in DNA. Uracil is in RNA. So cytosine, guanine are in both. So is adenine. The difference is thymine and uracil. DNA has thymine. RNA has uracil. So if you see a code and you see U's in it, you know you're seeing an RNA code. If you see a code and you see T's in it, you're looking at DNA. So, as we said, each nucleotide has a phosphate group, has a pentose sugar, and that differs depending on which you're looking at, RNA or DNA. Slight difference. The ribose for DNA is missing an oxygen that would normally be found in the whole ribose sugar. Why do they call it a pentose? Anyone know? It's a 5-carbon sugar. And it looks like a little penthouse. So how many bases are there, nitrogenous bases? 5. There are 5. But there's only 4 found in each DNA and RNA because there's a difference. So as we said, thymine only in DNA, uracil only in RNA. So notice again too what we talked about earlier when we were looking at that little quiz. The purines, when you look at their structure, they have 2 rings. You see that? So your purines are A and G. So I have another saying for that. Right? Oh, I forgot it. Oh, isn't Ag on the periodic table gold? Yes. Yes. Ag. So angels are golden. It's all about the angels. Ag is gold. And then the others that you're left with are the pyrimidines, so just have a single ring. So anybody other than adenine and guanine is a pyrimidine and has a single ring. So phosphate group, so it's a phosphate with 4 oxygens, and they tend to pull electrons, they lose protons, and so they tend to be negatively charged. Compounds that do that are said to be acidic. Oh, there's a clue there. Okay, so here's our 2 sugars, right? So here's our DNA sugar, so deoxyribose. So here is our ribose sugar. This D here is referring to the 3-dimensional orientation of how it's arranged. So you could do a mirror image of this molecule in 3-dimensional space, and that would be the left-handed form that's denoted L. The most natural organisms, they have the right-handed form, but it's written D. I don't know why it's written D. Okay. So that's a little bit of organic chemistry for you. Isomers, right? Isomers. So don't freak out about that D. I don't know why they put it there. But they number the carbons. And so they start in a clockwise fashion. So here's the oxygen. But here you have 1 carbon. At this point, they don't have it drawn in. 2, 3, 4, and then 5. And so these are numbered, and they put a prime symbol, that little dash, on them, because they number also the carbons in the nitrogenous bases. And for some reason they numbered those first, and so they put the prime on these guys. So when you compare this molecule to this molecule, do you guys see where the missing oxygen is, which carbon? It's the second one. So see this one? There's an OH right here, where there isn't an O here. That's the only difference between these 2 sugars. So here's it blown up. So remember, A, G, C, or T, right? For RNA, what do we have?
A, G, C, U.
Yep. A, G, C, or U. And then at the second carbon, you see that it's missing the oxygen here where ribose has it. So now, when a sugar of one nucleotide bonds with a phosphate of another nucleotide, these molecules, this oxygen and this hydrogen, are lost. They actually form water. So they stack up on each other. So this bonding, though, how they talk about is related to the carbon. So notice that the oxygen that interacts with the phosphate is on the 3' carbon, where the phosphate group is attached to the 5 carbon. This is where we get directionality for these molecules, because it goes from 5 to 3, from 5 to 3, from 5 to 3. Same is true, of course, for RNA. Right, so when we form strands, right, they're going to be missing these because that's where they form the bonds with each other. So some other terminology. When we refer to just the base in the sugar, it's referred to as a nucleoside, right? So no T, an S instead, and that S is for the sugar. So an adenine plus a ribose would be an adenosine. Again, sign referring to that it is a nucleoside, not a nucleotide. If it is the deoxyribose, they would say that, deoxyadenoside. Sign. When you have the base, the sugar, and the phosphates, or phosphate, it's referred to as a nucleotide with a T. You probably have never even heard of nucleosides before today, huh? They exist. For a short period of time, nucleosides do exist. We actually have enzymes to break apart nucleosides, to be able to break apart the base and the sugar. So some nucleotides you may have heard of are adenoside monophosphate, and this one just has a single phosphate. But we can add another phosphate, and so this becomes diphosphate, or the most common other abbreviation other than DNA and RNA is ATP. That's adenoside triphosphate, where it actually has 3 phosphate groups. ADP and ATP are used in our cells for what? Energy currency, right? The energy it takes to add those extra phosphates on helps store that energy. And when you remove those phosphates, you release that energy. Okay? And so this is a very important, common, what we refer to a lot of times are modified nucleotides. Right? Some of them will say that this is modified because we don't normally, when we're building DNA and RNA, we don't usually add more than 1 phosphate. Anyone know another one that's commonly used as an energy carrier other than adenine? What's the other abbreviation? It's guanine. If you ever see GTP, right? So the difference is the base, right? That's the only difference there. So, right, at the nucleoside level, you just have the nitrogenous base, and the sugar, right? And then at the nucleotide level, typically you just have one phosphate. But if you have additional phosphates, right, usually up to 3, then it's denoted by either di or tri, right? And a lot of times for a singular nucleotide, they will specify that it's just a single phosphate by writing monophosphate. And so that's where we get the abbreviations that we know of AMP, right, ADP. Now know what these stand for. So now let's go to the strand level, right? So that's the individual nucleotides, right? Let's put them together to form a strand. So we got to connect those sugars to the phosphates. That bond, that specific name for that bond, is a phosphodiester bond. And as I pointed out earlier, right, those happen at specific carbons as it relates to your sugar molecules. So the direction is 5' to 3'. And that connection of the sugars to the phosphates, is commonly referred to as the sugar-phosphate backbone, right, of DNA. This is very important to the structure of the DNA. It is very much like our very own backbone, right? It provides support. It really isn't the information, right? What carries the information in DNA and RNA? The nitrogenous bases, right? That's the code. That's the information. But this backbone is just as important, right? Because you have to be able to be a structurally sound molecule, right? And be stable. And believe it or not, this is a pretty beautiful, stable molecule. So we hook them, right? Stack them one on top of the other, right? So here's the 5' sticking out, right? Nothing's attached to it. And the 3' of that sugar is what's attaching to the phosphate, which is attached to the 5' carbon on that sugar. And they just keep stacking on top of each other. The nitrogenous bases are sticking out, right, from the backbone. This is important because enzymes will read in this direction, right? They go 5' to 3'. Right? They literally can tell the orientation of these molecules, and they follow that direction. So not only does it help us in orienting ourselves when we're looking at a code, but it'll help us understand how that code would be read too. So this we commonly refer to as the leading strand, right? Because this is the direction in which the RNA polymerase goes, or DNA polymerase goes, where the other strand goes in the opposite direction, right? So that one is commonly the lagging strand because they can't just go down it, right? They've got to build from the other direction because the enzymes only go in that direction. So that's actually the reason why we have the leading and lagging strands, right? It's the directionality of the molecule, right? And how the enzymes actually read it. Why didn't they just make one that could go the other way, huh? Why don't we have both? 5' to 3' and 3' to 5'. We could just zip down the whole thing wicked fast, but it doesn't happen. So 5' carbon is where the phosphates attach, right? The 1' carbon is actually where the nitrogenous bases attach. So there were several key experiments, and this is when we start getting into Watson and Crick, right? And the whole debate about poor Rosalind, right? On how they looked at her data, supposedly without her knowledge, you didn't give her credit, yeah, for how paramount looking at her data was at helping them figure out this overall double helix structure of DNA. So she died before they were awarded the Nobel Prize. She worked in a lab with another guy, which was Maurice Wilkins, and he also got credit along with Watson and Crick. I think he shared part of the prize, but she died before the prize was awarded. So they don't give it to dead people, do that. The sad thing is, and I looked this up because last night I was curious when I was reading over. Anyone know what type of research she did? Yeah, she worked with X-rays, right? So she took X-ray pictures, and she was pretty amazing at what she did, again, in that time frame. And those pictures that she took, that they looked at, really helped them in figuring out that it's a double helix. And how many turns were actually within that helix was based on the pictures that she took using X-rays. So yeah, unfortunately, she died of ovarian cancer. And I wouldn't put it past the fact of that she was not protected during her research, right? What she was doing, she was not protecting herself and basically irradiated and killed herself. She died at 37. Really sad. So that's an interesting story for sure, of the pains of research for sure, right? There's a bunch of books that have been written in all kinds of things, and probably even a movie about it. So I'm going to table this one for next time too, so read up on it if you're not that familiar with Watson and Crick and how they came about this overall beautiful structure of the double-stranded DNA. So we have Chase and Hershey to talk about, right? The bacterial viruses, and then these guys. Try to get some of those terms better cemented in your brain now.